Good morning, everyone. A warm welcome to all of you in this room and everyone who is joining us virtually today. It gives me great pride to share that we have over 826 attendees from 30 countries that include people living with ALS, their loved ones, as well as representatives from over 140 pharma biotech venture capital firms, over 135 academic institutions, and over 46 nonprofits that are part of this unique gathering today. And together, you represent the Target ALS ecosystem, its collective strength, and its ability to have an exponential impact so that we can accelerate towards realizing our vision that everyone with ALS lives. And it is only by working with you that we are going to succeed in our mission. So as we continue to expand our ecosystem and grow in momentum, I want to share with you briefly the unique holistic approach we are taking to accelerate the best ideas for ALS research from anyone, anywhere in the world. So our approach has three strategies. So the first is research we fund. We want to support the best ideas on ALS research while continuing to insist that scientists work collaboratively to advance their ideas. And our focus will remain on understanding the fundamental biology of the disease, helping accelerate translation of ideas from lab towards the clinic, and discovery and development of biomarkers. And we're excited and looking forward to hearing about the progress made by Target ALS-funded projects over the next few days. Second is research we enable. By providing access to critical research tools and resources to scientists from academia and industry worldwide. These resources range from simple reagents like antibodies to precious human biospecimens. They continue to be accessed quickly and with no reach-through on intellectual property. And we are really proud that over 1,650 projects have used these resources already, and these numbers continue to grow every day. We have also added new resources, and you'll hear about some of them from my colleague Amy Easton shortly. Third is research we conduct. This is a new area of focus for us. And the goal here is not to generate intellectual property for the foundation or publications for our science team. The goal is to help the research community address long-standing intractable challenges that have faced the ALS research landscape. One of the initiatives we have ongoing is a transformative global natural history study, which is creating the most diverse and comprehensive set of longitudinal biofluid samples and multi-omic data sets from them for the worldwide research community. You'll hear about this from Amy as well. I encourage you to also check out the poster from my colleague Laura Dugam, who's doing an incredible job leading this effort, along with the lead PI, Bob Bowser, and all the investigators who are part of this initiative. Now, before I go, I want to thank our board of directors and members of our independent review committee who are a source of wisdom for us. I also want to acknowledge the relentless dedication of my colleagues on the Target ALS team, who always focus on the mission first. And it is my privilege to be able to work with them. And now, I would like to turn it over to the source of inspiration for me, my colleagues, and imagine all of you, our founder, Dan Doctoroff. Thank you.
Thank you. Manish. I can't talk very well, but to do wonders of it, I prepare. Short, speech, and here it goes.
This has been a difficult year. At last year's Target ALS Annual Meeting, I got a cold and it developed into pneumonia. While I was in the hospital, my pulmonologist recommended that I have a tracheostomy. My wife, Elisa, and I made the decision to have it immediately. I spent 16 days in the hospital, and when I came out, I needed aids 24/7. My privacy was lost, and I was in a 430-pound wheelchair other than I was asleep. Over the course of the year, I've been losing my voice. That's why I am using an AI application called 11 Labs. And my legs, arms, and hands have weakened. I know that's the nature of the disease, but I'm an optimist, and there is nothing more than I'm optimistic about than ALS research and Target ALS. Over the past 3 years at Target ALS, we've increased our research budget by 4 times. Over the past year, we've made amazing progress at Target ALS. Our values are impatient optimism, deliberate disruption, and radical collaboration. We collaborate with everyone, academics, companies, other ALS organizations, other neurological disease organizations, and the NIH. We fund, enable, and conduct research projects. As the largest private funder of ALS research in the world, our consortia grants are fueling the future of ALS treatment. We are also engaging new scientists. Uniquely in the field, we provide a suite of critical tools and resources that empower scientific innovation. We have enabled more than 1,650 projects. We venture into the unknowns of ALS, conducting studies, including a global natural history study that explored the disease in diverse populations across the globe. We will soon have 18 sites across the world. In the past year, our independent review committee has decided to fund 21 projects out of 177 applications. 470 principal investigators applied from 254 institutions and 26 countries. In early 2024, thanks to Amy, Easton, and the science team, we established a data engine, which 300 users have already used the data engine. I'm especially proud that in 2025, Target ALS is funding 3 new research consortia focused on developing novel modalities, approaches that target ALS at the genetic level. These projects explore advanced tools like CRISPR gene editing and antisense oligonucleotides, technologies designed to precisely turn off or correct the harmful genetic instructions that may drive the disease. Each consortium brings together top academic researchers and biotech partners working side by side to move these complex therapies from the lab bench toward real-world clinical use. We are making progress on basic biology, genetics, biomarkers, AI, and novel therapeutics. As I said, I'm an optimist. I will adapt to whatever comes my way. I'm overwhelmingly happy. When I was diagnosed, I realized there are only 2 things that matter in life: relationships and purpose. In the past year, Elisa and I have had 3 granddaughters born. That makes 5 granddaughters born since I was diagnosed. They all live in Brooklyn, and we live in Manhattan, so I get to see them often. I have relationships starting with my family, who have been amazingly supportive. I have a ton of friends. As for purpose, nothing is more important than everyone living with ALS. That is my mission and my purpose. All of you in this room and on video are contributing to the rapid advancement of ALS research. We are going to solve this disease for every ALS patient. I have to thank Manish Raisinghani, our CEO, since Target ALS was founded in 2013, our amazing team, our board, and the independent review committee. I will continue to be involved in Target ALS until I can't do it anymore, which I hope will be for a very long time. Thank you.
Good morning. I'm Amy Easton, Senior Director of Scientific Programs for Target ALS, and it's my pleasure today to talk to you about the scientific strategy and mission for Target. As you know, ALS is considered a rare disease, and yet there are at least an estimated 400,000 people today all around the world living with ALS, and 1 in 400 people have a lifetime risk of developing the disease. The average age of disease onset is 55, but the disease can affect people much younger, even in your 20s. And once diagnosed, the average life expectancy is just 2 to 5 years. So with those statistics, it's somewhat disappointing that we still have only 3 treatments with limited effectiveness or limited application. And if you look beyond the borders of the US, even fewer people around the world have access to existing treatments or access to clinical trials. And our mission is to change this story. Through that, we're going to be removing key barriers that slow down progress to catalyze development of new effective treatments for people living with ALS. So we've identified key barriers that span 3 areas of research. The first is the study of the fundamental aspects of the disease biology. This is not one disease with one cause and one cure. There is patient-to-patient heterogeneity in the root cause and in the cellular and molecular events underlying motor neuron loss and clinical heterogeneity. So we need to remove those barriers to allow scientists to facilitate a deeper understanding of the disease biology. Second, there are barriers to key steps in the drug discovery process. Barriers to entry into the drug discovery pipeline is challenging without the right tools and human data to support nomination of new targets. Access to tools are needed to develop drugs that cross the blood-brain barrier, and access to the right preclinical models are needed to demonstrate drug activity. And third is the area of biomarker discovery. Biomarkers are critical for early diagnosis and to ensure optimal clinical trial design. Biomarkers must be non-invasive, so we need to rely on surrogate biomarkers to tell us what's happening in the brain, and we need to understand what those biomarkers mean. They must be specific so that we can distinguish ALS from other types of diseases, and they must be sensitive enough to detect low levels of disease markers in the blood. All of this work takes time. And so to tackle each of these key barriers and to accelerate research, we are applying a holistic strategy of funding multidisciplinary consortia, providing hard-to-get core resources with no strings attached, and through the research we conduct, generating comprehensive deep characterization of ALS on a global scale. So our funded portfolio reflects our commitment to advancing research in each of these 3 key areas. You can see in the figure here that about 1/3 of our portfolio is dedicated to understanding the basic disease biology, another 1/3 is dedicated to supporting drug discovery, and another 1/3 to developing biomarkers. And across these 3 areas, our research portfolio is tackling key biological targets that includes TDP-43, C9orf72, and FUS. And we're also tackling emerging themes in disease biology, such as the role of innate and adaptive immune systems in motor neuron degeneration. So with over $23 million invested across 62 projects supporting 153 scientists, we are excited to be accelerating new discoveries, many of which all of you will hear about this week. So although ALS is complex, there are unifying aspects of the disease. In particular, pathological aggregates of the protein TDP-43 accumulate in the brain of 97% of people with ALS. And under normal conditions, TDP-43 regulates RNA processing and the production of RNAs and proteins that perform fundamental cellular activities. But in ALS, TDP-43 becomes mislocalized, aggregates in the cytoplasm, causing disruption of many cellular events, including RNA metabolism, and leading to the formation of so-called cryptic exons that you'll hear more about this week, disrupted transport of RNAs and proteins, and other changes that can contribute to degeneration of motor neurons. Given the critical role of this protein, we are focused on helping the community study TDP-43, spanning basic molecular biology to research to discover TDP-43-directed diagnostics and therapeutics. In addition to funding this research, we're also complementing that by developing a really nice suite of tools for the community to use. And so these tools span preclinical models that are available, as well as well-characterized antibodies against TDP-43. And I specifically want to thank Len Petruccelli for generating an antibody to phospho-TDP-43 and enabling us to distribute this. This has already made a huge impact on research and having been distributed to almost 100 projects so far. This antibody has also been used in a large digitization project being led by Brent Harris at Georgetown University to help us fully characterize TDP-43 across 300 different cases, allowing scientists to understand TDP-43 in great detail and to do this with more rigor. So with these collective efforts, the hope is that in the not-so-distant future, an individual may be able to take a diagnostic test for ALS even prior to first symptoms to give patients the option for early treatment and prevention. So the postmortem tissue has been used to study TDP-43 and the pathological hallmark of ALS, but the insight we have gained from postmortem tissue goes way beyond this. So since our early days as a foundation, we've invested in a postmortem tissue core, and it's a comprehensive resource of both samples and data sets that we've curated for the community to allow researchers to study the disease in cellular and molecular detail and to dramatically increase the speed of new discoveries to understand the disease biology better. And part of our goal is to make those samples easily available and freely available. The core is being co-led by Dr. Harris and Dr. Bowser, and is supported by an amazing team of scientists and coordinators across 6 institutions. Together, they're stewarding the largest tissue repository for ALS with 530 cases of well-preserved and well-characterized tissue. And on average, we fill 50 requests a year. So this is supporting enormous amounts of important research all around the world. And there are at least 100 publications coming from the work done with this tissue. In addition, this year we are generating new data sets. We always continue to expand and grow them to allow you to do more work today than you could yesterday. And we're applying state-of-the-art techniques that allow you to see things that you would not be able to see with more traditional techniques. And so some of the data sets you're going to see in the data engine are long-read whole genome sequencing, spatial transcriptomics, and also, again, phospho-TDP-43 digitized images. And later this year, you'll see single-cell transcriptomic data being added to the data set. Again, we're really thrilled to be able to provide this resource to the community to support basic biology and drug discovery and biomarker development. So I want to now switch to our newest effort to talk to you about AGRI, our global research initiative to study ALS. Consistent with our mission to break down silos in research, we've launched a global initiative to study people living with ALS of all ethnicities, races, and underrepresented communities. AGRI consists of 2 complementary clinical research studies. The first is a global natural history study that Manish mentioned earlier, which is purposely designed to collect longitudinal data from people living with ALS to better understand disease progression and biomarkers of disease progression and the heterogeneity that exists amongst those individuals. And the complementary study is a more simple, streamlined study that's in community. So we go out to the community through a one-time pop-up event to allow people to participate in research who might not have otherwise. And together, the studies will enable really a comprehensive data capture from approximately 6,000 diverse people living with ALS over the next 5 years. And so now what I'd like to do is I'd like to introduce to you more about the natural history study, but I thought it would be better for you to hear from some of the voices of the people who are actually leading the study. And so I'd like to play this video for you now.
Target ALS initiated a large study to help generate the data and the biosamples that are really necessary to help fuel and accelerate research in ALS. We're going to learn so much from the inclusion of a multitude of populations from disparate countries with increased ethnic diversity. So I'm really excited about the future in ALS research because of this study.
Hace 19 años tenemos un grupo de trabajo para abordar los problemas de las personas con esclerosis lateral amiotrófica y sus familias. Estamos vinculándonos al esfuerzo importante que hace Target ALS para incluir varias poblaciones en sus procesos de investigación y construcción de conocimiento desde diferentes poblaciones que nos aportan una variabilidad genética, una variabilidad epidemiológica que probablemente nos permita entender mejor la enfermedad y cómo afrontarla. Gracias por esta oportunidad.
대한민국 루게릭병 환자들은 서양과 유전적 배경이 다르며 발병 연령이 더 젊은 반면에 C9 리피드 익스펜션이 드뭅니다. 이러한 차이는 질병의 다양성과 치료 반응에 영향을 줄 수 있습니다. 따라서 이번 글로벌 연구 참여가 더욱 의미 있을 것으로 생각합니다.
Tenemos mucho interés en poder caracterizar nuestros pacientes, al igual que poder entender cuál es la patofisiología que está ocurriendo en los pacientes, dado que no todos los hispanos son iguales, hay mucha heterogeneidad y es importante evaluarla.
So we, particularly from NIMHANS, we have studied the disease progression of ALS and the clinical and demographic characteristics, and we have highlighted that Indian patients do have an onset of the disease at a much younger age, and they do have a slower course of the disease, and they do live longer, almost for a mean age of around 6 to 9 years, depending on the onset site.
Israel offers unique genetic characteristics that are invaluable to ALS research. Our large family sizes, high consanguinity rates in select populations, and genetically isolated communities provide rare opportunities to identify genetic factors in ALS. These characteristics must be studied now before future generations become more genetically diverse. At Hadassah's ALS clinic, we maintain a comprehensive database with extremely detailed phenotypic data, genetic profiles, and also collect potential environmental factors. This creates an ideal foundation for the Target ALS Global Natural History Study.
So many thanks to those folks for sharing their messages, and please feel free to connect with them. They're all here this week for you to talk to and find out more about their work. But now I'm going to talk to you about where we are with this particular study. We've got 229 participants enrolled. 106 have already completed the study. Of those 229 participants, we have 84 healthy controls enrolled and 145 people with ALS. These individuals are enrolled at 14 different sites. You can see on the maps there. A number of them are US-based sites, but also international sites, including Colombia, Israel, and Korea, along with India, Malaysia, and South Africa. Those are sites that we're onboarding this year. We've done a really amazing job of collecting 23,000 vials of biofluids, blood, CSF, and urine longitudinally from individuals who participated in the study. And we've opened the doors to allow these samples to be used by industry and academics who've been actively doing biomarker discovery, and they're presenting their research here this week. And we've been generating comprehensive data sets from the study. So we've been making amazing progress, and it's really thanks to the participants for enrolling in this study. So I just want to share with you a few bits of data from the study. The first is that in terms of the cohort that we have enrolled, 64% are white, of European descent, but amazingly 46% of the rest of the individuals are of other races and ethnicities. So we are on track to meet the goals that we had in mind to create a diverse, rich data set. In addition, we have sporadic and familial forms of ALS represented with 44 individuals with known genetic mutations. Some of those individuals are non-symptomatic, and some of them are symptomatic who've been enrolled, with another 114 with no known genetic cause. And then finally, I want to mention that you can see in the figure on the right that if we were to plot data from participants who completed the study, you can see that we're capturing disease progression across the 5 visits that we're collecting data from. So participants come into clinic every 4 months for 5 visits. And you can see with the red line that we plotted data from 59 mutation negative ALS individuals, and we plotted their ALS FRSR scores over time. And we're capturing the progression that occurs in these individuals over a 16 to 18-month period. The green line is showing data from 8 C9orf72 repeat expansion carriers. Again, you can see that we're capturing their progression over time, perhaps a little bit more accelerated than the average group. And we'll find out more as we add more data to that particular set. And finally, we have, represented by the blue line, 16 SOD1 mutation carriers. And what you'll notice is that the ALS FRS scores are pretty flat and stable throughout the time period. And these participants also happen to be mainly on tofersen. 15 out of 16 of them were on treatment. And so we're happy to see that this data set is doing what we intended to do. It's going to provide a rich set of samples and resources for all of you to study disease progression and to understand the differences between different subgroups of patients, from their genetic heterogeneity to their ethnic and racial diversity. Finally, all of these data sets are already available in the data engine. So many of you have already checked this out. More than 300 people have accessed the data collections from the postmortem tissue core, a natural history study, and our stem cell collection. And so we're really excited to share that even new data sets were uploaded last week, including long-read sequencing, and we anticipate proteomic data to be uploaded in the next month. And this is really thanks to our partners at Somagen, New York Genome Center, and also the Zetterberg lab at the University of Gothenburg. And thanks to DNA Stack for being amazing partners for hosting the data engine, and to Verily Workbench for supporting this platform to help mine the data. So with that, I'll just wrap this portion up and say thank you to all of the people with ALS who've been participating in our studies, and thank you to all of you here today for advancing science. We're happy to work with you on this mission to build a world where everyone with ALS lives. Thank you.